Hello, everyone, and welcome back to the episode 11 from the Transfusion Squad. So, today we will be discussing and showing you a very basic and a primitive method of uh, checking for the leucoreduction status in the various blood products. So, Newbur's chambers and Nageotte's chambers, you must have heard these names. So, Newbur's chambers was used in our physiology, which we have already done in our MBBS times. And a similar platform called as Nageotte's chamber is used for counting low concentrations of cells and hence used to detect the limit and the level of leukoreduction in the leukoreduced blood components. So we'll be taking you through the steps, and I have Dr. Prachi Janoti, who's a first year resident with us, and she'll be showing you how to calculate the leukoreduction from using a Nageotte's chamber. Thank you.
Nagotte's counting method describes a procedure for visual counting of leukocytes present in leukodepleted blood or packed red cell products. The sensitivity of this method is zero point 0.1 leukocytes per milliliter and should be used for procedures in which leukodepletion has reduced the count to levels below 5 leukocytes per milliliter. The Nagotte's counting chamber has a volume of 50 microliter because its total grid area is 100 millimeter square and its chamber depth is 0.5 millimeter. The volume is calculated by multiplying area into depth. Hence, the volume is 50 microliters. The materials required for this experiment are Nagotte's counting chamber with a cover slip, a petri dish, normal saline, Turk solution, 100 to 1000 microliter pipettes, pipette tips, plastic tubes, a laboratory microscope. This is a packed red blood cell bag which has been prepared by leucoreduction using an inline leucoreduction filter. Gently shake the bag and mix the content using a stripper. We take an adequate sample of the bag using a sealer. The segment contains an ideal sample of the bag representing the content of the bag. Now we will be using a hematite segment device to collect a sample from the tubing of the packed red blood cell bag which we selected to conduct the experiment. Gently twist the tubing on the hematite segment device to collect the adequate sample in a clean test tube. Next, we will pipette 320 microliters of Turk's fluid in a clean test tube. Next, we will pipette 80 microliters of the sample into the tube containing 320 microliters of the Turk's fluid. Rinse the pipette several times to mix the two fluids until the pipette tip is no longer coated with intact red cells. This is a Nagotte's counting chamber. Place it on a moist petri dish to prevent evaporation. Place the cover slip on the Nagotte's chamber. Then carefully load the diluted sample into the Nagotte's chamber, ensuring that the chamber is completely filled. We will place the loaded chamber in a moist environment for about 15 minutes to allow the leucocytes to settle on the grid. Place the Nagotte's chamber on the microscope and using a 10x objective, count the white cells present in the entire 50 microliter volume of the counting chamber. White cells appear as intact cells that are refractile with a blue-gray color. Calculation. WBC concentration is expressed as C is equal to N divided by V into K, where C is the concentration of WBC per microliter, N is the number of WBCs seen in the chamber, V is the volume of the chamber, which can be 50 or 100 microliters, K is the dilution factor. Here, dilution factor is amount of sample taken divided the total amount of sample plus Turk's fluid, which is 1:5 dilution factor. Here, the number of WBC seen in the chamber was 1. The volume of the Nagotte's chamber was 50 microliter. Hence, the concentration is 0.1 WBC per microliter. Hence, adequate leucoreduction is achieved as verified by Nagotte's chamber.
Thank you, Prachi, for taking us through, and I'm sure everyone must have revisited their MBBS days and recollected the new burst chambers as well. So, to proceed ahead, can you tell me what is the composition of the Turk's fluid which you have used, and what is the function of each component of the Turk's fluid?
Thank you for the question, ma'am. The composition of Turk's fluid is 1 to 2% glacial acetic acid, which acts as red blood cell lysing agent. The dilute acetic acid in Turk's fluid is hypotonic to red blood cells, causing them to swell and burst, thus eliminating them from the sample. The dye gentian violet or methylene blue. It stains the nuclei of the white blood cells, making them prominent against the cleared background for counting, and distilled water, which serves as a solvent for the other components.
Right. So, can you also describe what is the major difference between the Newbar's chambers and the Negotte's chamber, so that it will be easier to understand and to compare and contrast both the procedures?
The new bar chamber has a volume of 1.5 microliter, whereas the Nagotte's chamber has a volume of 50 microliter. The new bar chamber is a single chamber slide used for counting cells, while the Nageotte chamber is a specialized double chamber slide with two independent counting areas for simultaneous analysis of two samples or cell types. Newbar chamber has a single standard grid, while Nageotte chamber has a double ruling for enhanced precision. Newbar chamber is commonly used for routine cell counts, while the Nageotte chamber is used for specialized applications requiring high accuracy.
So, using these chambers is quite cumbersome and a lengthy procedure to detect leucoreduction, and using it very often is not quite feasible in day-to-day practice. So, could you also let us know what are the other various ways of detecting leucoreduction, and how do you practice for detection of this during the routine QC of various blood components?
Thank you for the question, ma'am. Other methods of detection of residual leukocytes are impedance based analyzers, Coulter principle. A dilute sample is passed through an aperture with an electrical current and cells passing through change the current's impedance, allowing for cell counting and volume measurement. Optical analyzer, flow cytometry or laser scatter. A laser beam passes through a flow cell, illuminating cells one at a time. The light's scattering and transmission patterns provide information on cell size, granularity, and internal structure, allowing a differential count of white blood cells. Flow cytometry is an accurate and objective method used to detect and enumerate residual white blood cells in leucoreduced blood products to ensure quality control. By using fluorescent dyes, which stains the DNA in nucleated white blood cells but not platelets, flow cytometry offers superior accuracy, precision, and sensitivity.
So yes, right. Thank you so much, Prachi, and I hope this has given some insight into the primitive methods and the procedures which were earlier used, and it is still used in fewer centers where the advanced facilities are not available. So if you have any questions, please comment them in the comment section below. Thank you.